So we're going to discuss current knowledge of SLE pathogenesis, and we're going to discuss interim results with newer agents in clinical trials, and several of these have not been FDA approved. And then we're going to discuss emerging hypotheses regarding the microbiome and lupus pathogenesis. So this is a very simple figure, which all of you no doubt understand with first glance. And we can see a number of mechanisms and agents that are arrayed. Here we have our T cell here at top, interfering with co-stimulation by an agent like abatacept, while successful in rheumatoid arthritis, failed in trials in lupus. Looking at anti-CD19, that has not done well. At Proteasome recently, they reported their interim results in which it failed. Rituximab, although very effective in rheumatoid arthritis, actually did not do well in the both lupus nephritis and non-nephritis trials, so it failed, although I'll point out that there is a third-generation agent that is now going into trials. It's FDA approved for the treatment of leukemia, and it's much more powerful with regard to the binding affinity for CD20 as well as for the Fc-mediated antibody-dependent cytotoxicity, so we'll see what happens. So this is actually rituximab and ocrelizumab, the second-generation anti-CD20 agent. That failed as well, although it's moving forward right now in the treatment of multiple sclerosis, but that's another talk. Now, belimumab has a circle around it. That's an antibody to BLyS, or BAFF, B-cell stimulating factor. We're going to discuss that in greater depth. And that actually was the first agent that was approved by the FDA based on randomized clinical trials, period. The only one that's ever succeeded the gauntlet, and quite remarkably so. There are a number of other stages and potential mechanisms that can be relevant and that are being explored. A different blocker for BAFF also interferes with APRIL. Atacicept actually took TACI, which is a receptor off our own cells that mediates this signaling. They fused it to a IgG constant region. They thought if belimumab is good, this should be better, and trials to date have failed, so not doing very well. And then there are a number of other pathways that feed in that are being evaluated, and the roles of macrophages and the idea that neutrophils, really fascinating data coming from Mariana Kaplan's lab and others, discusses the contributions of neutrophils. Neutrophils, when they're triggered appropriately, actually spew their internal organs out into the battlefield, and in fact they create what are called nets, which are strands that are of host DNA arrayed with antibacterial molecules, and some of the molecules that are in this net trying to kill bacteria are actually immune enhancers like adjuvants. And there's a really fascinating article that says that these nets, a lot of that DNA may come from mitochondria. In fact, that's interesting because mitochondria are kind of like our symbionts. They have their own DNA that is used to synthesize not all, but many of the mitochondrial proteins and enzymes involved with oxidative metabolism. And that DNA is actually more bacteria-like. It's about 15,000 base pairs, and they think that that may actually be more immunogenic, and it's part of the way that a triggering of a neutrophil for netosis may be a very effective way, sort of a landmine to induce bacterial death. But the point is, in the context of a autoimmune disease, and whether that's lupus or whether or not that's actually ANCA-associated vasculitis, it may enhance autoreactivity. Interfering with those pathways are being examined. And we're talking about over here how there's involvement of immune complexes that can be enhanced by a plasma cell generation over here, antibodies, deposition of complement, and this onion-looking structure is the glomerulus, which can be damaged. So these are the many pathways that have failed, but there are still opportunities. I have to go from right to left, okay. I just wanted to bring up something that I hope you're all aware of, because there was a new chapter in the progression of our understanding of pathogenesis, and it relates to the idea of type 1 interferon. What is the role in lupus pathogenesis? And it's historical, going back to 1979. That was actually first shown that patients with lupus, more so than other healthy individuals, have something in their serum that causes the death of virus-infected cells. Later in the early 80s, I love the 80s, it was shown that this was actually interferon-related compounds, and that in fact, conversely, as we developed agents emulating interferons, we're infusing them into patients that have hepatitis or malignancy, looking for some clinical responses. We found by introducing the simple compound, we could induce lupus-like syndromes in many patients. So it seemed to fulfill some of Koch's postulates. Going on, looking further at patients' blood cells, several different groups simultaneously seized upon recently developed technology where you can, in fact, extract RNA from total blood cells and you can hybridize them onto arrays and you can figure out which are the most frequent transcripts in one group versus another. And what they found was that most lupus patients had what they call an interferon signature. They really couldn't figure out which type of interferon was inducing it, but it could be very, very remarkable in rays, very abnormal, made you think there are endogenous viruses or something going on to stimulate these, the white cells in the circulation of these patients, but reinforce the notion that there is a dysfunction or a dysregulation in type 1 interferon that's either the cause of lupus in some patients or the effect, but not completely clear. So looking at this, people thought maybe that's a good target. And also there was a linkage shown between interferon and actually induction of BAFF or BLyS, that survival factor that can interfere with tolerance mechanisms and result in expansions of autoreactive B cells, as I discussed yesterday. So just look a little bit closer at how interferon is linked to BAFF. Here you can see a very simple pathway where interferon released from another kind of cell, a plasmacytoid dendritic cell stimulated by DNA molecules, can actually stimulate a myeloid dendritic cell to make lots and lots of BAFF. And that can interfere with tolerance mechanisms in B cells and result in a cascade that is self-reinforcing, differentiation to plasma cells of autoantibody producing nature, and these will actually interact. Many of these antibodies bind to apoptotic particles, all triggering complement and all resulting in activation of further dendritic cells, plasmacytoid and myeloid. So it's a self-perpetuating cycle involving a number of the other factors that we've just discussed, and actually seems to be very important for pathogenesis, but the question is, how can you interfere with this cycle? Let me just teach you a little biology. This probably looks more intimidating than it is. Just what I point out is that type 1 interferon, although this signature is seen in the bloodstream of about 40% of adult lupus patients and more than 60% of pediatric patients, it looks like something's triggered the receptor for alpha interferon. We don't really know what it is. It's actually been a bit of a challenge, in part because there are many type 1 interferons that share limited homology, and all these many different factors, and this is how many copies they have within the chromosome, it all interact through the same receptors, alpha interferon receptors 1 and 2, and this is distinct from other types of receptors. The type 2 receptor, interferon gamma, as a separate receptor, but the point is that when induced, it can induce a very similar signature as the alpha type 1 interferons. So it's not so straightforward. Then there's a lambda interferon, just so to confuse everyone, and we really don't understand what the role is either. But again, they can have a common outcome. So one of the first approaches to try to interfere with this cycle was the development of antibodies that would bind type 1 interferons. But I just told you that this is very problematic because there are many different type 1 interferons. Many are structurally different than each other. And early trials have been reported the year before last at the ACR, and this is an agent called sifalimumab. And when they use different composite indexes for clinical outcome having to do with whether or not you can have a change of 4 points. This is somewhat similar to a SLEDI, changing your SLEDI 4 points or 6 points or 8 points. And here comparing it to a placebo standard of care, patients get a lot of steroids. It looks like treating with sifalimumab provides some benefits. And of course, more benefits if the change is less, 4, 6, or 8. But this is not so impressive, and I have to say even more disconcerting, and makes you wonder what is going on, is that there's no dose response. In fact, the more of the agent that you give doesn't really change, in fact, the clinical outcome. As we can see, there are 3 different levels of the drug. But it suggests something was going on. I should also point out that the interferon signature did not change dramatically in the patients that had high interferon signatures. Something was happening at the low end, low hanging fruit. People with low signatures, you could blunt a little bit. Didn't seem like it was a very powerful agent. They waved their hands. They said, maybe the dose isn't higher. And I waved back and said, yeah, but higher doses aren't doing more. I don't know what that's supposed to mean. So generally well tolerated, but in fact there was some problems with herpes zoster. And you go, does that make sense? A virus that lives within us, treating something that blocks interferon and then the virus more commonly activates? It's like, yes, it does. So is that going to be a problem in general? Well, let me just point out again, this is the pathway. Interferon alpha goes into these very specific interferon alpha receptors, and then reminding us that many of these pathways involve the JAK family, and in fact we have JAK1. The members of the family are JAK1, JAK2, JAK3, and type 2. Somebody's going to explain that to me today, what's with that, but here this receptor involves type JAK1 and type 2, and this will result when it is activated in the transcription factors going in the nucleus and having a result that's pro-inflammatory. So the next level of agents that were developed very logically was to interfere with the receptor. Ligands are too diverse, antibodies were not as effective, although amongst great hopes. And so the newest agent and the earliest trials were presented at the most recent ACR meeting is an agent which is called anifrolumab. I'm bad at these things. All right. So we'll just call it ANIF or something like that. And this is against the receptor. It's a monoclonal antibody. They looked at patients with moderate to severe lupus, which is really part of the unmet need. Well-designed trial. Patients received infusions every 4 weeks, were followed for 48 weeks, and they actually segmented those patients by whether they were interferon high or not. They were looking at transcripts in a interferon signature, 21 different genes, standard kind of approach. And I have to say this is very complicated to look at, but what I'm going to just cut to the chase, an easier slide, multiple doses done. Problem was that they actually had too much influenza that seemed to have a dose-dependent increase in the frequency, as well as zoster. The signature is coming up again. And here it's really pretty simple to see that patients with an interferon high signature compared to the placebo response rates basically had seemingly significant increases, 23% response rates, not too much of a jump after that, and actually went down with the higher dose of this drug. The interferon low patients actually got no results that were greater than the placebo itself. So it suggests that this may be a relevant mechanism because patients actually had significant benefits, and they in fact had drops of their composite scores of 4 or more. So something to be further evaluated and seemingly more effective and more biologically effective than targeting the cytokine itself. To shift gears, it has often been said that rheumatologists are proud of their subspecialty, but they're not too proud to borrow agents from other subspecialties. And this is a case where trials have been done with a class of agents called proteasome inhibitors. So this was an interesting cell biology observation in search of a clinical application, but really remarkable. Proteasomes are protein complexes that are inside all eukaryotic cells, and their main function is really to degrade unneeded or damaged proteins by a proteolytic process. I think of this as sort of the garbage disposal of the cell. So you don't need this anymore, you make too much of it, the protein's misfolded, you just put it in the hopster and push the button and it goes away. So this is a very important function because otherwise you'd be filling each cell with debris and useless material, and you wouldn't have the building blocks for useful proteins. And there are some cells that are really just at the razor's edge between functioning and being bogged down in their own waste. And the thought is that cancer cells are often like that, but so are plasma cells that are the antibody-producing factories of the body. They're just secreting and secreting and secreting. This is a simple demonstration. Here is our proteasome set up as this cylinder, and if you plug it up with bortezomib, which has been approved for the treatment of myeloma, but not any of our rheumatic diseases, this cell just fills up with its own waste and goes pop. I could hear it. It's specific but reversible, and cell sensitivity varies by type. Some cancer cells are very, very sensitive. They just basically have a limited tolerance for excess production. You clog up their waste button and that's it. And myeloma cells, this is a godsend. So many rational approaches to the treatment of myeloma failed, but it turned out that since myelomas are just producing so many antibodies, that this actually has been very, very effective, although there are certain neurotoxicities associated with it. So in Germany, there was a study that was done looking at patients. First, there was studies in mice that actually suggested it was a good idea, but then they moved to patients, 8 lupus patients with refractory lupus nephritis, got 4 21-day cycles of bortezomib. Again, this is not approved, it's off-label, and they found remarkable benefits. SLE scales, the SLE disease activity scale, on average, the patients had an average scale value of 13, which is very high disease activity. They had active lupus nephritis. This treatment regimen, they dropped to 4, which is really a very impressive response. Similarly, there was drops in anti-DNA antibodies. I have the data on the next slide, but at a significant level, and seemed preferentially compared to protective antibodies like the anti-tetanus antibodies here. So looking at other measures, a surrogate for interferon actually seemed to be decreased as well. And complement levels returned towards normal. The other point was that, in fact, if you looked at whether the effects were reversible, within several days of turning off the medication, 10 days, in fact, the plasma cell levels started to return to their pre-treatment levels, so this is not going to necessarily be an irreversible effect, and if there are some complications, it may well be, you can deal with it. So this is just to show you the kind of data. Here, the SLE rapidly over time are dropping, meaning that the patients are doing better clinically. And on the right, we can see anti-DNA titers are going down. So these are very important for showing clinical improvements. There also were some improvements, although less impressive on this graph, in proteinuria. So this is, I think the parental compound bortezomib may be too toxic. It's thought that the boron itself contributes to the neuropathy, but other analogs are being developed. But there's more complicated immunobiology. In fact, proteasomes, which are present in all cells, there are 2 flavors at least. There's a constitutive kind of proteasome, and then there's something called an immunoproteasome because it's more prevalent in immune cells, and it has different subunits. How do you know that? They're in yellow. They're very different. See, they're not blue. And if they make a compound that has higher intrinsic specificity for the immunoproteasomes, the goal is that perhaps we'll even be able to have greater benefits interfering with certain cytokines, and particularly more relevant for diseases that are immunologically mediated. And so this is a simple representation. One agent for a macrophage may be able to knock down all of these different cytokines involved in different phases of the autoimmune response. In fact, the point is that to accomplish this by biologic agents, you'd have to use a number of different agents, and so you get more of a broad response. I find this very ironic that we start out in an era where we use things like glucocorticoids and methotrexate that do lots of things and may interfere with many different pathways, and we go, no, we're going to go to a smart kind of system, pinpoint bombing of individual molecules and cells, take them out of the equation, it'll be a much better effect. And they go, oh, well, maybe we didn't get in all patients the greatest level of efficacy, so we're going to actually figure out agents that, in fact, screw up many of these circuits. But in this case, the JAKs will inhibit it by the wiring inside the cell, and this is by turning the waste accumulation against the cell itself. So there may be new opportunities. Here, this also interferes with T-cell generation that are involved in regulating the immune response or dysregulating something that turns into an inflammatory response. Take-home lesson from this is that an approach is under investigation for treating inflammatory and autoimmune disease by specific targeting of immunoproteasome subunits. And this may have less toxicity and affect a greater range of immune cells, and a very clever approach. A minor segue to finish my program. I have been deeply immersed in the microbiome. All of us are, whether you know it or not. And this is I'm immersed, usually in the laboratory, though. And so this is a picture from a review issue in Nature, and what it seeks to demonstrate is that although we think we're humans and we know what we look like, that in fact we really have this other self that really is a composite community of many different kinds of bacteria, archaea, and viruses. And in fact, may be more important in our day-to-day existence and our health and disease than most people recognize, but it is essential to our being. And as I like to say, we all contain a zoo, and who doesn't love a zoo? So our intestines in particular have many, many different bacteria in it. It's the home of 10 to the 10th to 10 to the 14th microbial cells. I was telling you how many lymphocytes we have. We have more bugs. And in fact, there are 1,000 or 2,000 different species of bacteria that are in our intestines, a very complex ecosystem. I find it astounding to think that for every one human cell, and we think we're special, and every one of our cells we think is special, we have 10 bacterial cells. And so we're really composite entities going through life. The question is, who's dominant? I'm not sure. But even more importantly, for every one human gene we have, our bacteria have 100. And you realize that it is an ecosystem, and it's very dynamic, and it can shift in response to stress, different nutritional intake, and it is much more adaptable. It doesn't have to mutate genes and have variants that take decades and decades. You can actually shift your microbiome within days to adapt to new influences. And I have to say, if we think about environmental factors, studies of genetics for most of our diseases have only shown contributions and susceptibilities. Maybe 20% of the risk of getting disease is from just the right mix of wrong genes. What else is contributing? We think environmental factors. You always point to tobacco and smoking, a lot of inflammation, evoking susceptibility to overt disease. Well, the bugs in your body are the largest environmental exposure that there is, period. The other part that's fascinating is that most of the bugs in your gut you can't even culture, so it's hard to count them if you're going to put them on a plate. They do not like oxygen, and they actually don't like to be separated from their friends. So the question is, how do we inventory them and see if there are differences in what they're doing for your metabolism? We'll point out that there are at least 3 major functions of the intestinal microbiome. They synthesize vitamins essential for health. They break down the food that we eat, processing complex carbohydrates, amongst other things, making simple compounds that, like butyrate and single-chain fatty acids, and breaking down to amino acids like tryptophan, and all of these can actually influence immune pathways and maintain regulation or their absence to interfere with immune regulation. And in fact, there are examples in which particular bacteria that are just part of what lives in us or our close evolutionary species like mice, something that can live there happily is not really a pathogen. If it's present early in development, can absolutely shift your entire immune responsiveness and set you up with lots of Th17 cells, for instance, which may be good for defending from infection if you're challenged, but if you have the wrong genes, they can trigger autoimmune disease. So there's a difficult balance, and it may all be determined in your bowel. And here we have an example that, in fact, different mixes of different bacteria will induce host responses with exuberant here and less exuberant host responses, depending on what the mix of bacteria that are present. And so whether or not this is all a setup for autoimmune disease, well, one of the most remarkable breakthroughs in technology was how to catalog the microbiota that are present without actually touching them too much and shifting their representation or culturing them at all, and it all relates to advances in next generation sequencing. So we can just extract all the bacterial DNA all at once, and we can actually use a polymerase chain reaction that's to a conserved part of a little gene that is essential for life, and there are parts of that gene that actually are invariant between all bacteria, but between a place where you put your two primers, it's highly diverse. So you can look at that sequence, and I can actually assign to any one of the different phyla or families or individual species based on that one gene sequence. It's not a complete characterization of the bacteria, but it's a great way to putting into several thousand different bins and looking at the relative representation, all based on just DNA amplification. And this will allow me to, in fact, figure out what the community looks like. I could subsequently go from phylogeny and to go on to take the DNA and put it through a different kind of amplification and look at, in fact, what the metabolic genes are to get a very good picture. So over time, there's increasing databases to figure out what's going on. I'm going to quickly show you some unpublished data that hopefully we'll take out of these slides before they go on the web. One of the first measures of how different your bacteria is the Chow-1 alpha diversity measure, and this is measuring operational taxonomic units. The concept is whether the sequence in the 16S is one sequence or another and relates to something that's close to a quasi-species. And so the Chow-1 just says, do I have a lot of species or I don't have a lot of species? Health generally correlates with a diversity of different species. They can all find a balance. They can all give each other what the group needs. But there have been examples in which different diseases, like inflammatory bowel disease, shows a contraction of fewer species. And there are less policemen that actually keep things in order. But the question is, what does this have to do with lupus? Lupus is not a bowel disease. Lessons where there is local inflammation, maybe you would think that there's a local imbalance. So these are our data, and this actually shows that healthy people show a range of diversity in different species in their bowel. On average, healthy people, and these are actually all adult women that seem like a reasonable comparison, have about 1,200 what look like species based on this characterization. Lupus patients have significantly less, so that was not particularly expected. The part that was particularly exciting is if we divided the patients up based on SLEDI scale, SLEDI low looked more like healthy people, and SLEDI high looked more messed up. And so these are cross-sectional studies, but it gives you the impression that perhaps as the disease progresses, and we have to look individual by individual, the distortion becomes more severe, the complex ecosystem that we need for health breaks down, and then in fact, we'll see that not only are there fewer species in general, what kind of bacteria are there? And the idea is, what we found is that they shift. So this is sort of a way of taking all bacteria in each of these individual points as an individual, and on average, what does their community look like? It's a way of estimating all the bacteria in your gut in one vector in 3 dimensions. And it turns out that some of the patients look like healthy people, but others are widely divergent. And this is if you see in 2 dimensions, and this is if you can see in 3 dimensions. And I really can't see that at all myself, but this is what the people who do this for real, this is statistically significant differences. But if you compare healthy people to, in fact, low disease activity to high disease activity, what you find is that they get more dissimilar from each other. So not only do they have fewer and fewer bacteria, but they are weirder and weirder divergence and expansions of bugs that are not really representative of healthy people. So you're getting really out of whack from an equilibrium perspective. And I'll just point out, if you can see this, we defined exactly the bacteria that were present in these groups of healthy people, low disease activity, and high disease activity, and found patterns for individual groups of bacteria that were statistically significantly different, looking closer and closer and closer. And we found several examples where bugs that maintain health in this case, this Prausnitz species has been cited to mechanistically induce T regulatory cells. They make the balance. Under their influence, everything is chill. And unfortunately, compared to healthy people, if you have a touch of lupus, you have less of them. And if you actually have severe lupus, you have less and less and less. And so the protectors in the bowel are being diminished. And similarly for this other protector, uniformis, it's decreasing and decreasing and decreasing. And then there are some species that have been implicated in the pathogenesis of other diseases, and primarily inflammatory bowel disease, which appears to become more and more prominent. And so we're in a certain ecological space, a niche within your bowel. You should, in health, have many different bacteria all working and playing together. Something happens where the diversity collapses and one bad actor expands, and we're going to isolate this exact bad actor from lupus patients based on their signature and figure out just what it wants to do and how it may contribute to a pro-inflammatory state. Okay, and this is looking at patients one by one by one for this potential pathologic factor. It is a commensal, so it's there in health, but it has the potential, we're speculating, to cause imbalances, and so a symbiont with pathologic properties. Sounds very science fiction. You can see that it is not very prevalent in healthy people, which is each of these over here to the left of the vertical bar, but lupus patients are heterogeneous, but some of them have very great expansions. And at the same time, there are other factors which are expanded in healthy people that seem to be reduced in lupus patients. And so it is the yin and the yang of the bowel, and we're losing our balance. But we have candidates that we'll bring in and interrogate severely. Okay, so what have we reviewed today? That there are many targeted approaches in lupus that have failed to meet primary and secondary endpoints. Alpha interferon has been implicated in lupus pathogenesis. Recent trials with antibodies that target interferon proteins have shown limited biologic activity, but actually the most exciting news is that the first studies with an interferon alpha receptor shown greater biologic activity, and we're hopeful that it'll be seen in further trials to have its place in the armamentarium, although not yet approved. Borrowing proteasome inhibitors that are accepted therapy in multiple myeloma in a small trial of lupus patients have encouraging results, although not yet approved. And we discussed the microbiome in lupus, that our early DNA sequencing technology is providing evidence of imbalances that may well be directly linked to disease pathogenesis. A number of other diseases we're finding similar changes. I didn't go through some of the vasculitis data, but they're looking for whether or not bugs may be more prevalent, being part of that pathogenic process. All right, so lupus patients commonly display dysbiosis in their intestinal microbiome with reduced diversity, and certain species become expanded more so than healthy individuals, and the idea is it appears the worse you are, the more imbalanced you are. All right, so now we will test your knowledge. Okay, so which of the following is an antibody to interferon alpha receptor? Is it A, atacicept B, blisbamod, C, sifalimumab, D, ocrelizumab, or E, anifrolumab, which I can't say very well. What do we vote? We vote that it's coming. Yes, yes. You've all, or most of you have selected the correct unpronounceable antibody. Ding! Okay. Now, this is really important, so when you go home, you'll have something to discuss in cocktail parties. Compared to the number of human cells in our bodies, how many bacterial cells are in our intestines? A, the same number, B, 10-fold, C, 100-fold, D, 1,000-fold, or E, too many do count. What do you think? Inquiring minds want to know. How many bacteria on the head of a pin displacing those angels? And the answer is tenfold. But I can't count that high either, so I would have accepted that answer. All right, last and perhaps least, which of the following is not a true statement regarding interferon and autoimmune pathogenesis? This is what a question such as I'm well known for, where it becomes so complicated that even the writer gets confused. At least that's what I'm told. Type 1 interferons include 14 family members, including interferon alpha, beta, gamma. I don't know what that is, kappa and squiggly. You know, in my home, we speak Latin freely, but... I think it's Greek. It's Greek, okay. No wonder I don't get it. Yeah, who was the vice president who said that when he went to Latin America, he wishes he could speak to people in their native language? So interferon, let me get on with it. Interferon alpha contributes to pathogenesis in many adult lupus patients. Well, we hope, or we think. Okay, interferon alpha infusions into hepatitis C patients and oncology patients can induce a lupus-like disease. D, the receptor for interferon alpha involves the Janus kinase 3. E, interferon-mediated activation can result in raised BAFF BLyS levels. So think about it. Assume you're betting your house, and you don't have another one. Yeah, that was correct. Okay, thank you so much. That's great.